Good evening, everyone. So it's already 7:15. I think we have waited enough for everyone to join. Uh, so we will start today's live session. I am Amrita, a PhD scholar at University of Hyderabad, and I will be taking your live interactive sessions for the course Industrial Biotechnology for the next 12 weeks. So today is week 1. So we will have a brief introduction of who all have joined in the class today. So before that, let me say something. So I hope you all have gone through the lectures given by Professor Devabrata Das and have joined the session today. That is a mandatory process before joining the live session, that you should have been gone through the lectures. Okay, without that there is no point of joining this live interactive session if you have not gone through the lectures. So I think already week 1 and week 2 quiz is available in the NPTEL portal. So these sessions are being conducted for you all to practice some extra questions which are not included in the quiz assessment, right? So those quiz assessments are given for your practice, and they are uh, having marks, but this live sessions doesn't have any marks, but it will give you a kind of revision of what you learned in the lectures every week. So I hope I'm clear with the goal of why we are going to have these live interactive sessions. So we are going to have it every Tuesday for the next 12 weeks from 7 to 9. However, whenever we finish the class. Okay, so every week we are going to discuss a set of 20 questions if possible. I have set the number to 20. If I can make 20 questions, it's going to be 20 or maybe less, depends on the type of topic discussed in that particular week. So, but I have set my mind that I will try to prepare 20 questions at least for each session. It can be a little bit more if the topics are more. Today, I have prepared 20 questions for you all, and these all are MCQ based questions, no subjective questions at all. And I will show you the question, and I will give you around 2 minutes time to think which of the answers can be correct out of the given four options, and whatever you think is correct according to you, please type the answer in the chat box. Okay, I hope everyone knows how to use the chat box in Google Meet. And please, the first and the foremost rule for attending this live session is do not uh, unmute your audio and video throughout the class. Okay, so please, as there are many people in this class, I don't want any kind of chaos. So it will be better if we keep yourselves unmute throughout the class, and I should be the only one speaking in this session. Okay, and if you have any kinds of doubts regarding the session or the questions, you can freely type it in the chat box. I will be continuously seeing the chat box, so no need to worry about that. Other than that, I think there is no need to talk. And yeah, so can I get a thumbs up or yes or some kind of answer that uh, you are able to listen to me and understand everything I have told you till now? Okay, great. I got a lot of thumbs up. So we will start. Is there any doubt before we start the session? Then just let me know. I think I'm clear. Don't raise the hand. You can just write down your query in the chat box immediately. Okay. Ashraf, has raised the hand, so please let me know what is your query. And yes, I will request you all to please join the session every day by every Tuesday by 7:10. Today I waited a much longer time because this is our first session, but I won't be waiting every Tuesday like this. So I will wait for 10 minutes. That's the maximum I can wait for you all. After that, I have to start the session. Okay. But in case you are missing the session due to any kind of emergency you have, for that reason, we have been asked to record these sessions. So I am recording it and I will upload it in the YouTube channel as well. You will get the YouTube link through the NPTEL website. Okay. So yeah, I think that is done. So no need of writing down questions or anything. You will get the slides, you will get the recording of the session. So no need to worry about it at all. Just focus on the revision. Okay. So is there anyone who has not attended the lecture and has joined the class today? Just let me know, or everyone has attended the lecture of this week 1. I'm just asking particularly about week 1. Is there anyone who has not attended the lecture? If not, then take it as a reminder from next week. Please come prepared, then only you will be able to answer the questions which I have prepared for you all. Okay, because everything is from the lecture for that particular week. Okay, so there won't be any outside questions whatsoever. So please be prepared. Okay, so I'll just repeat one more time that I will show you the question. It has four options. Think thoroughly which can be the correct option for you, and then uh, write down or type it whatever correct answer you think, and then we after 2 minutes I will show you what is the correct answer and what is the explanation. Okay, so the explanations are not going to be very detailed like I'm taking a class for you. I won't be making you understand the concepts now. I think that is already done in the lectures. Okay, we are just here to revise the with the questions. Okay, I will explain some briefly. Okay, so if you still want a detailed explanation or if you have confusion, you can ask me, but I think it won't be required because everything is from the slides. Okay, week 1 slides provided. Okay, so we will start. Some questions have detailed explanations, some doesn't have, which I felt is not required. I have not included it, but if you still want, you can ask for it in the chat box. Okay, so I think this much of introduction is enough. At the last, you all can give me an introduction. After we finish 20 questions, I will ask you all for a brief introduction like what are you studying? Are you doing BSc, MSc, or whatever? I will love to know how you all are and what kind of courses are you taking and what made you choose this course. What was the need? I will ask it. Okay, so let's share all those information but after the class. Okay. Okay, so we'll start the class. Everyone is ready, so I will show the first question. I hope the slides are visible. Okay. Okay, so we'll start with the first question. Which of the following is not a typical characteristic of an industrial stream? Option A, high product yield. B, long fermentation duration. C, genetic stability. D, use of cheap substrates. Okay, read the question properly and start answering in the chat box. You can just write A, B, C, or D. No need to write the full answer as well. Okay, so think and start writing. And please do not trust your co-fellow mates that if someone has given answer A, then I will also give answer A. Please trust yourself the most in this session and what you have learned in the lecture, and based on that answer it. Okay. Roshni Goyal has raised the hand. I would like to know what is the query, Roshni. Please type it in the chat box. And I can see 54 to 55 people have joined here. So I need 50 answers at least from everyone. I should see 50 answers coming for question number 1. Okay? And my only point of saying to be active throughout the class and to join the live sessions is you can practice with me live. Obviously, I will send you the recording and you can go through it anytime, but uh, that is every time a boring process to see the video and learn through it. And comparatively, it is always interesting to have a live session and get your answers and queries cleared immediately. Okay, so just because you're going to get the recordings, please do not skip the live sessions. Okay, because I have to also give attendance for you all. I have to update your attendance in the NPTEL team. So please keep that in mind and join the live sessions every week. Okay, only in some kind of emergency, if you have missed it, you can go through the recordings. Or also, if you want to revise the questions again near your exams, near the time of your exams, then you can go through the recordings or the slides again. Okay, otherwise, do not miss the sessions. Okay, I think many of you have given the answer. 2 minutes are up, I think. We'll see what is the correct answer to this question. It is B. Long fermentation duration is not a typical characteristic of an industrial strain. What is the explanation? Industrial strains are selected and engineered to maximize productivity within a shorter time frame. So, a longer fermentation duration increases the cost and risk of contamination. If you see, this was the slide given in the lecture. So, I have kept slides not for all the questions but for some questions. So, you can go through it, and if you really need the slide reference for any question which I have not kept, you can ask me. I will provide you in the next class. Okay. So these are the characteristics of industrial strains. What are these? Genetically stable and rapid cell growth, high rate of production of the desired product, ability to utilize wide variety of inexpensive substrates, shorter time of fermentation. So this was your question. It was given longer time, which is not correct. Fewer byproducts, which makes simpler downstream processing, ability to undergo genetic manipulation, and ready harvesting ability of the product. So these are the characteristics of industrial strains, and longer time of fermentation is not included in it because it leads to contamination. Okay, I hope this is clear. We'll move on to the next question. 1 minute. Just give me a sec. Just 1 minute. I'll show you the second question now. Yeah. So, yeah, I hope everyone can see the second question now. Okay. So, what's the second question? Which of the following strategies is least likely to enhance metabolite overproduction in an industrial microbial stream? flux rerouting for precursor optimization, overexpression of negative regulatory genes, metabolic engineering of biosynthetic pathways, removal of product feedback inhibition. Start answering. Okay. Has everyone done answering? Yeah, one more thing I want to tell you all. So, I need to know how many of you are giving correct answers and how many wrong answers you all are giving. So, I would like to know your score out of 20 at the end of the class. Okay? So, please keep noting down how many correct answers and how many wrong answers you all are giving. We will know your marks at the end of the session, like how much you got, 18 out of 20 or 90 out of 20 or 20 out of 20, whatever it is, just keep noting down. Okay, and there is no need to be ashamed of if you get less marks or anything. Just is a practice class, a revision class, only uh, aimed for your improvement. So yeah, be frank about it, and if you have scored less marks, you can always score a good mark in the next session. Okay, so yeah, I think I have given you all enough time for question number 2. So the correct answer is B, overexpression of negative regulatory genes. So this strategy is the least likely, you can say, strategy to enhance metabolite overproduction in an industrial microbial stream. So here you can see the explanation for it. Overexpressing negative regulatory genes typically suppresses the biosynthetic pathways, as those are negative, not regulatory genes, but negative regulatory genes. These can suppress the biosynthetic pathways, reducing metabolite production, right? Whereas the question asks least likely to enhance metabolite production. So obviously they can't enhance metabolite production, as those are the negative regulatory genes. Whereas flux rerouting, metabolic engineering, and removal of product feedback inhibition are all target higher yields. They all target higher yields or higher metabolite overproduction in an industrial microbial strain compared to the overexpression of negative regulatory genes, which is a wrong thing. Okay, so I hope this is clear. Moving on to the next question, third question. What is the primary reason for classifying citric acid as a low value, high volume product? It is produced in minimal quantities. It is highly expensive. It has low cost and high annual production. It is difficult to purify. Start answering. And if you need any kind of changes in the live session or any suggestions you have which you want to include in the live session, that is also most welcome. But it should be within the rules which I have been given to follow. If yes, then I can surely entertain it. Okay. What are these reactions coming for? I can't understand. I can see many people are giving correct answers, so I'm thinking that you all have attended the lectures. Obviously, you have joined the course, so you should learn and go back with something. So yeah, you should attend the lectures as well as the live interactive sessions. It's hardly 1 hour in a whole week. I think you all can take out time for it, right? And it will just improve your uh, skills on this course, right? So I'm just thinking maybe everyone is from life sciences background because I am from that. And if someone is from some other background also, I would like to know. We will discuss it at the end of the class. Okay. So is everyone done with third question? I need to see 50 answers for each question. I don't think everyone are giving answers actively. Some people have just joined to listen to the class but not to participate in it. But these are live interactive sessions. Interactive session means you have to participate in this, okay? So it's very boring to just sit and keep listening to me, right? Instead, you should be active and participate in the this. I have made it like a Q&A session so that it can be interesting for you all. Or else I could have just come and then I could have just sit for 1 hour and read the question and answers. There is no point of asking you all to say it. I know the answer already, but this is for you all so that if you have given a wrong answer for some question and then you rectify it through this live session, you will remember that answer till your exams. At least you will remember it till your exams. So no need to revise it again and again. Okay, so lot of talking is done. Let's see what is the answer. It is C. It has low cost and high annual production. Why? Citric acid is classified as low value, high volume product because it has a low cost and high annual production. So, the explanation is low value, high volume products like citric acids are inexpensive and required in bulk quantities for food and pharmaceutical industries. Their industrial significance lies in the demand, not high unit price. So, if we look at the slides given, here you can see this was the slide from which the question is taken. These bioproducts whose cost is less than 6 euros per kg and are usually required in large quantities, millions of kg per year, are termed as low value, high volume products. Example is citric acid. Citric acid is nothing but the acid we find in lemons, tomatoes, right? This is a very common thing. Factors which play a crucial role in fermentation of low value products: the cost of the raw materials, duration of the fermentation process, overall cost of the utilities. These are the factors the uh, which play a crucial role in fermentation of the low value products like citric acid. Okay, so I hope this is clear. Moving on to the next question. Question number 4. Which of the following is true for chemical processes but not for bioprocesses? Operate under ambient temperature and pressure, use of metal catalysts, require sterility, generate biodegradable products. Start answering. Fourth question is here. Read the questions properly and then answer. Okay. So you can please inform your fellow mates who have joined the course and have not joined the live session today that this is a very important session which you all should attend because attendance is being marked for these sessions, and yeah, they should not miss it until it's an emergency. I'm not sure how many people have joined the course. I don't have a number for it, otherwise I could have asked, but yeah. who all are joining and you know that your friends have not joined the session today, please inform them that from next Tuesday, please join. Personal questions in the sense, what kind of personal questions you want to ask? I'm not sure. Related to course queries. Okay. I'm not sure if I know the answer to all your personal questions, but shoot, let's see. What are the questions you want to know? Like, I am only assigned for the live interactive session. Other than that, I don't think I can answer any of your questions. But yeah, just ask me. If I know, I will tell you. Okay. Is everyone done with the fourth question answering? Let's see what is the correct answer. It is B. Use of metal catalysts. Which of the following is true for chemical process? So, use of metal catalysts is something done in the chemical processes, but not for bioprocesses. So, explanation, you can see chemical processes often use inorganic catalysts such as platinum or nickel and may require higher pressure or temperature. In contrast, bioprocesses use bio means biology, so obviously there are no metal catalysts required. Bioprocesses use enzymes or live organisms and are generally more sustainable and eco-friendly. So this is a common sense based question. Anyone who doesn't know also they can answer it. Okay. So I think it was clear. So here you can see why bioproducts, because they are sustainable, less carbon and water footprint, creates rural employment opportunities, less emissions to the environment, biodegradability and recyclability, high productivity, use of raw materials from the local sources. So these are some of the advantages of using bioproducts. Moving on to the next question. Which preservation method is best for ensuring long term genetic stability of an industrial microbial strain? Periodic transfer to fresh media, overlaying with mineral oil, lyophilization, freeze drying, saline suspension. So, start answering. Is the chat? Yeah, please ask your queries in the chat, not through mic. As I said, I don't want you all to unmute your mic and create any kind of disturbance in the ongoing session. So, whatever you want to ask, please ask it in the chat box. I am continuously seeing it, I won't miss it. Okay. And people who are joining late, please try to join on time from next Tuesday. Okay. Please do not miss the questions, it will only hamper your revision. 50 people are there attending these live sessions, so I need 50 answers for each questions. If you deduct me, 49. I need 49 answers from each for each question. Okay, and I don't think everyone is still answering. Obviously, I can't point out who is the one not answering here. So just be honest and answer it. Okay, I guess I have given enough time for this question. Let's see. The correct answer is C. Lyophilization or freeze drying. So, what is lyophilization? So, lyophilization using a cryoprotectant and airtight sealing can preserve cultures for decades, minimizing mutation rates and genetic drift compared with the growth based methods. So, this is something done to ensure long term genetic stability of an industrial microbial strain. So, people who are in research, they must be knowing this, right? Whatever, who all are working on bacteria or any kind of microorganism, they know that how do we store it. How do we store the strains? We store it in -80, that is the most commonly used temperature for lyophilization. At -80, if you store the cultures, so they will be can be stored for decades, and there will be minimum mutation rates and genetic drift compared with the growth based methods like periodic transfer to fresh media. Literally, how many days can you do it? Just transferring to fresh media. This can't be done forever and ever. Or else overlaying with the mineral oil. That also can't be done forever. Saline suspension. So these all are just temporary methods, but lyophilization is something you can use for a longer period of time. Right? Here you can see preservation of industrial strains. This was the slide. Industrial cultures must be preserved and maintained in such a way as to eliminate genetic change, protect against contamination, and retain viability. Based on their properties, techniques for the preservation of microbes are broadly divided into two categories: preservation of industrial strains, methods to keep the organism metabolically active. Right? So if you want to keep the organism active for a few days, you can keep transferring to fresh media. That is the first thing. Or overlaying culture with mineral oil, or storage in sterile soil, or saline suspension. These are the methods to keep the organism metabolically active. But if you want to store it in a submerged metabolic state, you have to go for these methods like drying in vacuum, lyophilization, or use of liquid nitrogen, or storage in silica gel. Okay, so these are for something you when you don't want a active metabolic state but a submerged metabolic state, right? So I hope this was clear. Moving on to the next question. Which aspect does not distinguish industrial strains from wild type strains? Superior tolerance to metabolites, higher genetic stability, poor adaptation to cheap substrates, high reproducibility of product formation. Start answering. Since the video is recorded 8 years before and the questions in the assignments are modern, so apart from the lecture, where can we see more such stuff? Since IIT exams are the toughest, what do you suggest? Okay, so yeah, so if you want advanced level lectures, I don't think I can provide you with it, but you may just use the internet to explore your level of thoughts for that. But questions are, if all the questions are from the lectures given, I'm not sure how much old it is. From you only I'm getting to know that it is 8 years old videos. But yeah, so I don't think there is any other source to get the same topics and same lectures. I don't think that only Professor Devabrata Das knows that. I don't think if the video, I didn't know that the videos are old. If they are old, then I don't think they are going to provide you with some recent um, videos or lectures. Or if the assignments are modern, I don't know which assignments are you referring to, the ones given in the NPTEL portal or the questions we are discussing. because these questions are made by me recently. And the assignment questions, I'm not sure how are those and on what level the assignment questions are made. But if you want to see stuff related to this, I'm 100% sure there must be a lot of stuff in the YouTube, or you can just ask AI for it. If you want to learn something more from the topics or covered in the lectures, right? I hope that answers your question, Vartika. Yeah, so yeah, okay, on the website. Okay. Yeah, obviously, assessment questions must be prepared every year, but the lectures are still the same. And if there are some current topics which needs to be included and they have been missed, yeah, we can't do anything about it. If you are really interested to know it, you can just uh, uh, like answer your curiosity through the internet. Yeah. So, yes, let's see what is the answer to question number 6. It is C. Poor adaptation to cheap substrates. So, which aspect does not distinguish industrial strains from the wild type strains? So, these are the options, and all the options qualify for the distinguished characteristics of industrial strains and wild type strains, except for option C. So the explanation is industrial strains are usually acclimatized for efficient use of cheap and varied substrate, unlike many wild types. Okay, so here if you see in the slides, this was the difference between wild type strain and industrial strains. So wild strains are having poor reproductibility, whereas industrial strains have high reproductibility in product formation rate and titer. Wild strains are susceptible to product inhibition, and whereas industrial strains are highly tolerant to product inhibition. Wild strains have poor genetic stability, whereas industrial strains have high genetic stability. And wild strains have the ability to use various substrates, whereas industrial strains are acclimatized to use cheaper and wild variety of substrates. Wild strains have poor substrate conversion rates, whereas industrial strains have very high substrate conversion rates. Right? So I hope this answers your question. Moving on to the next one. UV radiation primarily induces mutations by deleting genes, breaking peptide bonds, forming thymine dimers, de-inhibiting transcription. Okay. So yeah, start answering. I need answers from 51 of you. I can see 52 is the attendance. Okay, I can see answers coming, but I don't think everyone is still answering all the questions. I don't know where's the problem. Okay, maybe I should give you all more time. If you need it, just let me know. If I'm going fast, I can slow down a bit. Or if you want me to go fast, that also we can do, but I don't think I can go fast. Okay, there is only two lectures, right? Two lectures? Till now, there are only two lectures given. Ma'am, I didn't attend the last three lectures. I think, okay, week zero, if you count, maybe three, you can say it. But week zero, week one, and week two lectures are given in the website as far as I checked today. So, what if you have not attended it, you can go back, and attend it, okay? But next session, say come prepared, okay? Okay, let's see what is the correct answer. It is C. UV radiation causes adjacent thymine bases in DNA to dimerize, leading to replication errors and mutations. It's a common method for induced mutagenesis in microbial strain development. So, whoever is in the research field or is in microbiology field, they know this, that we always switch on the UV in the hood before work starting our work. So what happens with that UV radiation? It causes the adjacent thymine bases in DNA to dimerize, leading to replication errors and mutations. It's a common method, but this is not the question about it. So it's asking that what happens and how the UV radiation induces mutations. So UV radiations are not just for protection in the lab culture, but it can also induce mutations if you leave any of it in the UV, but we don't do it, right? It's a common method for induced mutagenesis in the microbial strain development. So if you see here, non-ionizing radiation, long ultraviolet radiation, it is a long ultraviolet radiation. They have low penetration power. The major effect of UV radiation is formation of pyrimidine dimers. You can see here what happens here. So the adjacent thymines, upon exposure to UV light at 260 nm wavelength, they become a thymine dimer. And this is what can induce your mutations. Okay, so here you can see the how it affects the genetic material before and after. Okay, this is the UV photon coming and what happens after that. So cytosine, and the cytosine thymine dimers are less prevalent. So always it's the thymine dimer. Okay. So I hope this is clear about the UV radiations. Moving on to the next question. Which of the following is not used as a carbon source in industrial fermentation? Molasses, cheese whey, urea, starchy wastewater. Start answering. Okay, is everyone done? I think this was a very easy question, doesn't need much time. It's a direct question answer. So yeah, it's answer C. Urea is not used as a carbon source in industrial fermentation. So what are the things which we generally use to induce fermentation? Those are molasses or cheese whey or starchy wastewater. So urea is a nitrogen source, not a carbon source. Okay, so carbon sources like molasses or whey supply energy and carbon skeletons for biosynthesis. Urea supports amino acid and protein synthesis, not energy generation, right? Okay, so these are what I was talking about. Carbon sources used in fermentation: molasses, cheese whey, malt extract, starchy wastewater, cellulosic wastes, sulfite waste liquors. Right? Moving on to the next question: which enzyme is correctly matched with its industrial application? Cellulase to detergents, isomerase to cheese making, rennin to cheese making, xylanase to dairy. Okay, start answering. Okay. Please keep yourselves mute throughout the class. As I told before, please do not unmute yourself. Yeah. Okay. Let's see what is the correct answer. It is C. Rennin is used to coagulate milk proteins in cheese production, not in cheese making, while cellulases are used in textiles or biofuels, and xylanases in pulp or paper. Isomerizes convert glucose to fructose and not used in cheese. Okay, so the answer should be B. One second. Yeah, is correctly matched. So all are correctly matched except for answer C. So the answer C is correctly matched. All are incorrect. Sorry for the confusion. So yeah. So cellulases are returned for detergents, whereas it is for textiles and biofuels, and xylanases are for pulp or paper, whereas it is written dairy in the question, and isomerases convert glucose to fructose, and it's not used in cheese, but it is written cheese making in the question. Yeah, so some things can be confusing if you don't read the question properly. Okay, so keep that in mind always. Okay, moving on to the next question. The Arrhenius equation expresses the relationship between pH and reaction rate constant and substrate concentration, reaction rate constant and temperature, pressure and activation energy. Okay, so start answering. Okay, is everyone done with the? Okay, people are still joining. It's 1 hour late. I don't know. Okay, so the correct answer is C. Reaction rate constant and temperature. So the Arrhenius equation expresses the relation between reaction rate constant and temperature. Okay. So what does it show? It shows that the rate constants increase exponentially with temperature, highlighting the sensitivity of reaction rates to thermal conditions. So with increase in temperature, the reaction rate constant increases. Okay, so that is what explains the Arrhenius equation. I'm sure there were a lot of numericals in the lecture, so I hope all of you have gone through it. I have not kept any numericals in this class. If you really want it, we can have it. So moving on to the next question. A batch fermentation for ethanol uses 100 grams of glucose and yields 51.1 grams of ethanol. The relatively low yield versus mass is best explained by non-optimal enzyme specificity, stoichiometry and byproduct formation, presence of heavy metals, contamination by foreign microbes. Okay, so start answering 11th question. We have more 9 questions to go. Okay, till now I have not received any kind of query. I need 51 answers. Maybe I will just start counting now how many people are answering and point out. So before I start doing that, please be active and answer it. Okay? It's okay to answer wrong question, wrong answers, not a problem. Whoever is raising the hand, please keep yourselves mute. Sapna, that's what I was saying, that please keep yourselves mute so that video and audio both should be mute so that there is no chaos in the class, and we can have a peaceful session. Okay, if one person is talking and everyone else is listening, that's a peaceful session. So whoever has a doubt, please write down what is the query in the chat box. I will answer it. Okay, Vimala, uh, if you are having any doubt, please write it down. Okay, I don't see anyone asking anything. Okay, so let's see what is the correct answer to this question. It is P. Stoichiometry and byproduct formation. So, a batch fermentation for ethanol uses 100 grams of glucose but yields only 51.1 grams of ethanol. So, this relatively low yield, because 100 grams was used and 51 grams was the yield. So, the relatively low yield is viewed can be best explained by stoichiometry and byproduct formation. So, the explanation is not all the glucose carbon goes to ethanol, right? Some is lost as CO2 or carbon dioxide and is used in cell biomass, reflecting the actual biochemical stoichiometry. Okay. So moving on to the next question. Which of the following is not a typical method for enhancing the yield of antibiotics in an industrial setting? Metabolic flux optimization, large-scale bioreactor scaling without strain improvement, removal of negative regulations, genetic recombination for desired alleles. Start answering. I hope my audio is clear. Okay, if there is any kind of disturbance and if you can't hear me clearly, just let me know. Okay. Okay, I hope everyone is answering. Let's see what is the correct answer to this question. It is B. Large scale bioreactor scaling without strain improvement. This is not a typical method for enhancing the yield of antibiotics in an industrial setting. So the explanation is scaling alone does not intrinsically increase yields. Rather, optimization and genetic manipulation are also required. Okay, like metabolic flux optimization, removal of negative regulations, and genetic recombination for the desired alleles. All these are required to enhance the yield of antibiotics, whereas large scale bioreactor scaling, it alone does not increase the yield without strain improvement. First, the primary thing is the strain improvement, then only you can opt for large scale bioreactor scaling. Okay. I think, okay, moving on to the next question. Which of the following applications does not employ industrial enzymes? Cellulases in pulp and paper, lipases in biodiesel production, tannase in wine fermentation, beta galactosidase in molecular cloning. Start answering. 13th question this is. No, obviously insulin is not an antibiotic, it is a hormone which regulates the blood sugar levels, right? What are antibiotics? You can just know it from the word itself. Something that kills bacteria. Insulin is not something that kills bacteria, right? It is a hormone produced by the pancreas to regulate the blood sugar levels in our body. Okay, so I hope that is clear. Okay, let's see what is the correct answer to this. It is D. Beta galactosidase in molecular cloning. While the question was which of the following applications does not employ industrial enzymes. So beta galactosidase is not something which you can say is an industrial enzyme. It is not used in industrial applications. Beta galactoside is something we use in our molecular biology screening processes. If someone is from molecular biology research, they know it. Whereas cellulases, lipases, tannase, all are having industrial applications, okay, as given in the question. So moving on to the next question, the difference between wild and industrial strains does not include genetic stability, tolerance to product inhibition, cell size, substrate conversion rate. Start answering. Please do not give two options as answers, just give one. Okay, if you are guessing also, just write one answer. B or C, you can't do that. Okay, let's see what is the correct answer to this question. It is C. Cell size. So differences between wild and industrial strains typically relate to genetics, productivity, and robustness. Cell size is not a selection parameter in strain development, right? So we have already discussed this. So nowhere cell size is given as a criteria to differentiate between wild strains and industrial strains. So that is the correct answer. Moving on to the 15th question, which process is associated with the transformation type of fermentation? Ethanol production, penicillin biosynthesis, amylase secretion, steroid conversion. Start answering. Next week, I hope to see more of you because, yeah, I am not sure how many of you actually thought we are going to have this session and which kind of session this is. So I have cleared all of you. So just inform your fellow lab mates, fellow mates, whoever are in the course, if you know anyone. Yeah, I am aiming for 20 questions. If you think it is too much, we can decrease it to 15 as well. But I think 20 is a good number to cover all the topics in each lecture. And yeah, you can have a good set of questions for revision as well. Because whenever you are going to have your exams, you are going to get a lot of MCQs in it. So if you are thoroughly practiced for each week with the MCQs given in the assessment and here, you will be prepared, I think. Is anyone having any problem with the 20? Because we are given 2 hours time. I don't think we are going to go for 2 hours. Today, there was a lot of discussion because it was the first session, so we started the class late. But from next time, I'm going to start the class sharp at 7:10. Okay, so please everyone be on time and we will try to cover the questions as soon as possible. Okay, but it can't be less than 1 hour. At least we have to uh, have this session for 1 hour. Okay. Let's see what is the correct answer. It is D. Transformation fermentation modifies an added substrate, example steroids, via microbial enzymes. It's not about producing microbial biomass or primary metabolites, but about bioconversion. Okay, so the correct answer is D. Steroid conversion is the process associated with the transformation type of fermentation. Moving on to the next question. Which biochemical pathway is involved in ethanol formation in yeast? Kelvin cycle, glycolysis, urea cycle, pentose phosphate pathway. Start answering. Okay, let's see what is the correct answer to this question. It is B, right? Glycolysis. In yeast, glucose is broken down via glycolysis to pyruvate, which is then converted to ethanol anaerobically. Okay, so ethanol formation is a part of an anaerobic process, not a aerobic process. Calvin cycle is for carbon fixation in plants. The others are unrelated, right? So here you can see the biochemical pathway of ethanol in the yeast. You can go through it, how glucose is converted to pyruvate, and then how it goes through a anaerobic process because of the release of CO2, and ethanol is produced. Okay. So moving on to the next question. Which approach is least suited for the preservation of fastidious, highly oxygen sensitive industrial microbes? Freeze drying, storage in silica gel, ultra freezing in liquid nitrogen, overlaying on mineral oil. Start answering. Okay, let's see what is the correct answer to it. It is D, overlaying on mineral oil. So this is the least suited approach you can do for preservation of highly oxygen sensitive industrial microbes. So mineral oil preservation is less effective for fastidious or strictly anaerobic cultures due to the oxygen diffusion. Ultra freezing or freeze drying, which we already discussed, is lyophilization, are preferred methods. Okay. So moving on to the next question, the presence of antifoam agents in fermentation medium is crucial because they prevent denaturation of enzymes, they ensure accurate pH control, they facilitate oxygen transfer and prevent loss of culture due to excessive foam, they act as trace nutrient sources. Start answering. This is 18th question. We have two more questions to go. Okay, is everyone done? Or should I wait for some more time? Okay, I'll just wait for more 30 seconds. Done. Okay. Let's see what is the correct answer. It is C. They facilitate oxygen transfer and prevent loss of culture due to excessive foam. So this is why anti-foam reagents are required in the fermentation medium so that they can facilitate oxygen transfer and prevent loss of culture due to excessive foam. This is the correct answer, which is C. So excess foam can interrupt oxygen transfer, contaminate air filters, or cause loss of valuable broth, so antifoams are added to control this. Okay. Moving on to the next. Which of the following best explains the use of molasses and corn steep liquor in industrial fermentations? They are required for sterility. They provide carbon and nitrogen respectively at low cost. They eliminate the need for antifoaming. They buffer the fermentation pH. Start answering. We have one more question to go. Then I would like to know what are your scores. Okay, so be ready for that. Okay, let's see what is the correct answer to this. It is B. They provide carbon and nitrogen respectively at low cost. So molasses are a source of carbon and corn steep liquor are a source of nitrogen. That's why they are being used in the industrial fermentations. So molasses supply fermentable sugars and corn steep liquor provides amino acids and nitrogen, making the process economical. That is at a low cost. So here you can see these are the nitrogen sources. Carbon sources we have already discussed. So nitrogen sources are corn steep liquor, ammonium salts, urea, peptone, yeast extract or soy meal. Whoever has unmuted, please mute yourselves. Everyone, please mute yourselves. Yeah. So moving on to the next question. This is the last question. Which of the following is a medicinal enzyme used for diagnosing myocardial infarction? Glucose oxidase, creatine kinase, cholesterol oxidase, trypsin. So yeah, start answering. Okay, is everyone done? Let's see what is the correct answer. It is B. Creatine kinase level rises in the blood after heart muscle damage, making it a clinical marker for myocardial infarction. Glucose and cholesterol oxidases are for metabolic diagnostics. So the correct answer is creatine kinase, which is a clinical marker for myocardial infarction. Okay, so that is a medicinal enzyme used for diagnosing myocardial infarction. So with this, we are at the end of our slide. So yeah, this was the slide I took from the lecture. So here you can see creatine kinase used in myocardial infarction. Creatine kinase activity is greatest in striated muscle, heart tissue, and brain. Okay. So yeah, other options were glucose oxidase, which is used for quantification of blood glucose level, and cholesterol oxidase for the cholesterol detection and quantification. So yeah, this was it for today. So yeah, if you have any questions, you can ask me only related to the live sessions. If you want to repeat any question or anything, you can ask. Or else, yeah, let's just, is there questions are taken from first week lecture? Yeah, these are the questions taken from the first week lectures. Okay, so let's see what is your score out of 20. No need to take screenshots, Vartika. You will be provided with the recordings as well as the slides. I will share the PPT with you all. You will get the Google Drive links for everything. So just wait for it. Okay. As I told, no need to note down anything. You will be getting everything. Uh, Next live lecture is again Tuesday. Next Tuesday, the timing and days are fixed for every week for the next 12 weeks. Already we have done with week 1. Next 11 weeks you are going to meet me every Tuesday 7 to 9 p.m. Okay? So I hope that clears the doubt. Anything else? Okay, so now let's look at your scores. Everyone, please uh, let me know your scores. Out of 20, who has scored how much? Please write down in the chat box. In week 1, no, I don't think I have gone through the lectures of week 1. This is what it was. I think that's what you're talking about, 0 week, I guess. 0 week was not asked for, so I have not prepared that. So, yeah, fastidious means there are microorganisms, like you can say bacteria, which need or they have some complex nutritional requirements or some complex environment or some specific environment which they need to grow on. So for that kind of bacteria or microorganism, it is used the word fastidious. Okay, so simply you can say some bacteria require specific nutrients in specific environment to be grown. So those kind of bacteria are called, or you can classify it as fastidious. Okay. Is there any question else, or you can just start with your scores? So fastidious is something which you have to work on or focus on. to grow it, not fitting in the standard laboratory conditions for growth. Okay. Yeah, anything else? Or you can all just start typing your scores in the chat box. Out of 20, I need 50 answers here. 16, okay? 14. Yeah, for someone who has not joined from the start, I'm telling again, please be on time from the next class, okay? 18 out of 20, very good. Okay, so there's no one who has scored full marks, 20 out of 20. Okay, all of you have scored good, 19 out of 20, very good, Chinnasamy. Okay, is there anyone else who wants to share their scores? You're most welcome. Okay, whoever has scored less than 15, please go through the lectures once more. Okay, go through the lectures and again go through the live sessions once more to rectify your mistakes and to focus on which parts you were not clear with and you need revision again. Okay, and who all have scored above 15, I think they are safe, right? Okay, so I don't think everyone has shared their scores, but it's up to you. I can't force you on it. So yeah, let's have a brief introduction of all of you. As I said, I am a PhD student. I am in my final year. I'm also a PMRF fellow, and I'm currently doing my PhD from University of Hyderabad. So I am in Hyderabad. Okay. So yeah, let's have your introduction if you want to give it. Like I really just type it, like what is your background, like current educational background. That's what I want to know so that I can imagine which level of questions I should make. Like, okay, I hope you got the point. BSc Bio 5th Sem. Okay. Anyone else? Okay. Masters in Botany. MSc Biotechnology. Good. So, okay. BSc and MSc, is it? BSc. Okay. BSc. BTech. Okay. You can write your institute name as well if you want to. BTech. Okay. So all are from biology background, I guess. Right? Okay. Okay. So most of you all are in your bachelor's, right? Yeah. Okay. Okay. Biotech. Most of you are in biotechnology BSc. I can see that. Okay. BTech or BSc biotechnology. Okay. So I hope. Okay, Piali. She's also PhD student. Microbial carotenoid production and all. I'm doing this course as my coursework. Okay. Okay, very good. Okay. Anyone else? Okay. Okay. Very good, Priya. Okay. Good, good to know you're working at a pharma company. Is these type of questions will ask in our exams? Yeah, obviously. That's why we are doing the practice, because you're going to get a lot of MCQs. I don't know the how many marks of MCQs you're going to get, because I have not been shared with the exam pattern or anything, but I know you are going to get MCQs. Those can be from your assessments which you are being given every week, or can be from the live sessions. It can be from anywhere, but it will be from inside of your lectures. So that is the most important part, that you should be thorough with. If you want, if you are giving an exam and if you are hoping for a certificate, then you should be thorough with it. Okay. Yeah, so I guess everyone is done with their introduction, whoever wanted to share, and the doubts are cleared. So shall we wrap up for today? um yeah. Okay, so we'll meet again next Tuesday, sharp 7 o'clock. Please. S- Mm, yeah, obvious. I think certificate will be given to all whoever have passed. Yeah, yeah. Thank you, everyone. We'll disperse for today and we'll meet again next Tuesday. Okay, please remember the timings. Next Tuesday, 7 o'clock. Please try to join on time. Do not make me wait. Um, I'm not sure about the passing marks, Vartika. Maybe you can write to the NPTEL team. They will help you with it. I think 30 to 40% will be the passing marks, I think. Yeah. Okay. Okay. Um, I myself was taking a NPTEL course in my MSc. So that time I don't think there was any kind of negative marking. Yeah, I don't think there is any. Whoever knows the answers to these kind of questions, they can just type it, because I am not aware of it actually. When I was taking a NPTEL course, I don't think when I gave the test there was no negative marking. Yeah, I don't know. As I said, I don't know the exam pattern, but I surely know there are going to be subjective plus MCQs. Okay, so subjective part, so you have to go through it through the lectures only, but MCQs. You can practice by me, through me, or through the assessments. Okay. Okay, so we are up for today. We are done. We'll meet again next Tuesday, everyone. Okay, so be on time, 7 o'clock. Okay, and please don't worry, I will provide you with the slides and the recordings, everything. Okay, so don't worry about that. How are you taking the attendance? I saw you all right. Yeah, Piali knows a lot, I guess. 100 marks exam will be there. 100 questions for 1 mark or 50 questions for 2 mark each. Yeah. You all can help each other. If you know, please share the information here. I don't know if you all can talk later or not. So if you have some doubts, ask here. If anyone knows, they can answer it. So that is also good. Yeah, someone asked something. Yeah, I will take the attendance. No need. I can see who all are here and I know the names and the classes are recorded. So I will take it. Okay. So don't worry about that. If you have joined the session by before 8, your attendance is marked. But after 8, I'm not sure if I'm going to mark your attendance or not. Because if you are 1 hour late to the session, I'm not sure if your attendance can be marked. Okay. So that's why I was saying please come or join on time. Okay. Okay. Is that all? Or if there is anything else you want to ask? Okay, then I will just leave the class. If you have any more questions, save it for the next Tuesday. Okay? Thank you, everyone.